Hello students, today we are going to discuss about a genetic material, that is DNA. In DNA specifically, today we are going to discuss about Watson's and Crick model of DNA. Okay, so the structure what we are going to discuss in detail is Watson and Crick model of DNA. So if you are looking into the structure of the DNA is a double stranded molecule. So how many strands are there? There are two strands. So you can find this is one of the strand which is moving. So this is one strand, right? So this is one strand and this one is the other strand. So how many strands are there? Two stranded, and they are right handed. So the helix, when you are seeing, they are somewhat right handed in nature, and they are double, that can also be clear. And another thing is that they are like a twisted ladder. So when you are looking into ladder, you know how the ladder will be like this, isn't it? So this is not like the straight one, it's a twisted ladder. Okay, so that is what the first point is dealing with. DNA is a two-stranded molecule, right-handed double helix, like a twisted ladder. Now each strand is having how many ends? There are two ends. What are the two ends? One is the 5' end and another one is the 3' end. Okay, so what is this 5' end and what is this 3' end? Listen carefully. So when we are drawing, we have drawn this is the DNA. So I am mentioning here 5', here it is 3'. One is moving from 5' end and ending at what? It is ending at 3' end. Another one is starting from 3' end, isn't it? And they are ending at what? They are ending at 5' end. What is this 3' and 5'? Listen carefully. So when you are looking into the structure of the sugar, if the carbon atom is means the nitrogenous bases are fixed to the which part? Third carbon compound. So if you are seeing the carbon structure, so this is the first position of carbon, this is the second position of carbon, this is third position, fourth position, fifth position, like this positions of carbon as well. Each and every bonds you can find what is present. Carbon is present. Glucose, if you are writing, how you will write? C6H12O6, isn't it? So here carbon, when you are seeing, if the sugar carbon is attached in the third position, then we call it as the third dash. 3'. What do we call them? We call it as the 3' end. And if the carbon atom to which it is attached, if it is in 5' position, then we call it as what? We call it as the 5' end. Okay, so it's based on the which position of carbon of the sugar the molecules are attached. Okay, if it is attached to the 3' third carbon compound of the sugar, then we call it as what? We call it as 3' end. If it is attached to the 5', then we call it as what? The fifth carbon atom, then we call it as the 5' end. Okay, so that is regarding your arrangement. So two ends are there, one is the 3' end and another one is the 5' end. Okay, and they are anti-parallel to one another. Anti-parallel means what? If it is from 5' and 3' and this strand is also from 5' to 3' means it is parallel, isn't it? Both are similar, same, parallel, but it is not like that. One strand is moving from 5' to 3' and another strand is moving from 3' to 5'. So these are not parallel to one another, they are anti-parallel to one another. That is the next thing you have to note. Okay, the strand which is having uppercase 5', okay, which is having 5' to 3', we call this strand as the sense strand. What do we call them? We call them as the sense strand. Okay, the strand which is moving from 3' to 5' end, we call it as what? Anti-sense strand. So one strand which is moving from 5' to 3', we call it as the sense strand, and another strand which is moving from 3' to 5', we call it as what? The anti-sense strand. Okay, so that one you have to make a note. Anti-sense strand, 3' to 5'. And when you are looking into the diameter of the DNA, what is the diameter of the DNA? The diameter of the DNA is it is 2 nanometer. How much it is? It is 2 nanometer. So NM is referring to what? The nanometer. So diameter is just an information. It's 2 nanometer. Distance between each turn is 3.6 nanometer. So which is each turn? Each turn means this is one turn, this is the second turn, this is the third turn, this is the fourth turn, this is the fifth turn. Okay, so the distance between one turn, so this is one turn from here to here, the distance is 3.6 nanometer. How much it is? 3.6 nanometer. Now this when you are looking into the structure of this DNA, the DNA when you are seeing, the backbone of the DNA is this one, the ladder. It is made, the backbone is made up of sugar and phosphate. What it is made up of? It is made up of sugar and phosphate. So this sugar and phosphate is the backbone, isn't it? And in between what you can find the nitrogenous bases. What you are having? Nitrogenous bases, adenine, guanine, thymine, cytosine are forming the ladders. Okay. So in each and every turn, so this is one turn, means in this turn, how many ladders will be present? Around 10.5 ladders. How many ladders? 10.5 ladders. So how many base pairing will be there? This is first base pairing, second base pairing, third base pairing, fourth base pairing, like that. 10.5 base pairing will be there. Approximately 10 base pairing will be there. Okay. So in each and every strand there will be around 10.5 base pairing. The distance between the two ladders. Okay. So this is the turn. In this one ladder to second ladder, the distance, when you are seeing the distance, is 0.34 nanometer. So what is the distance between the two? That is 0.34 nanometer. Okay. So each turn it is 3.6 nanometer. Distance between the two nitrogenous base pairing is how much? It is 0.34 nanometers. So like that, how many can be arranged? Around approximately 10 base pairs will be there in one single turn. Okay. So that is regarding your distance between each turn is how much? It is 3.6 nanometer. Distance between each base pairs is how much? It is 0.34 nanometer. So hence, how many nucleotides will be there? 10.5 nucleotides will be there. I am making it clear once again. If it is not clear, each strand how much distance? 3.6 nanometer. Between the two base pairing, how much distance? 0.34 nanometer. So like that, how many base pairs will be present in one turn? Around 10.5 nucleotides base pairs will be present. Okay. And another thing what you have to notice that there are two grooves present. Okay, one is the major groove and another one is the minor groove. The distance between the two nucleotide bases where it is more, so that one we call it as the major groove, and where it is very less, that part we call it as what? The minor groove. Okay, so there is a major groove and there is a minor groove. So what is the backbone of the helix made up of? I told you already, they are made up of what? The sugar and the phosphate. So when you are looking into, this is sugar and this one is phosphate. So this is the backbone. Okay, and what is the ladders made up of? The ladders are made up of nitrogenous bases. Thymine, adenine, guanine, you can find thymine, adenine, guanine, cytosine, thymine, adenine, thymine, isn't it? So that is made up of the nitrogenous bases. So backbone of the helix is made up of sugar and phosphate, and nitrogenous bases are of four types, adenine, guanine, thymine and cytosines. So nitrogenous bases are in turn divided into two, that is one is purine and another one is pyrimidine. Purines are who are? Adenine and guanine are purine, thymine and cytosine are pyrimidines, isn't it? And it has been noted that, listen very carefully, adenine will always pair with thymine. Adenine will be always pairing with whom? It will be pairing with thymine, and guanine will always pair with cytosine. So this is a purine which always pairs with the pyrimidine thymine. Guanine will always pair with cytosine. Okay. Now one more thing which you have to notice, that adenine and thymine, between them there are double hydrogen bonds. How many bonds are there? Double bond. Between guanine and cytosine, how many bonds are there? There are 3 bonds, triple bonds. Okay. So this sort of base pairing we call it as complementary base pairing. So whenever I am writing adenine, on the opposite side what will be there? It will be thymine. And wherever guanine is there, it will be cytosine. If thymine is there, then automatically it will be adenine, and when cytosine is there, automatically it will be guanine. Okay. So like that, complementary base pairing will be there. Adenine will always pair with thymine and guanine will always pair with cytosine. This sort of base pairing we call it as what? Complementary base pairing. And in this complementary base pairing, you can find always the number of purine will always be equivalent to number of pyrimidine. Okay, so number of purine, that is your A plus T, will always be equivalent to your G plus A, G and TC will always be equal. Purine will be equivalent to what? The pyrimidine. Okay, so that is another thing. And the Bonding between them is what? Hydrogen bonds. Okay, these all things you have to make it clear. So this structure we will just discuss. You can find it is the sugar and phosphate backbone, right? Nitrogenous bases are forming the ladders. Nitrogenous bases, adenine will always pair with thymine and guanine will always pair with cytosine. So adenine plus thymine will always be equivalent to G plus C. Okay, held together by what bond? Hydrogen bonds. So this is more clear. If you are seeing a 3D view, it will be looking like this. Oxygen base pairs are present in the ladder. Carbon of the sugar bond, thereafter phosphorus will be present. So this will be the somewhat the structure. Okay, so one turn if you are seeing that is regarding the same. Now moving on to the next one is your structure. Here you can find this is the nitrogenous bases, this one is the sugar and this one is the phosphate. Okay, this is the deep groove and this one is the minor groove. Okay, so that is again a thing. One more thing what you have to note is that purine, when you are looking into the structure, the purines are having two cycles, two rings are present. So you can find this is two rings, this is two rings, this is two rings and this is two rings. So adenine and guanine are having two rings. Chemical compound structure, when you are seeing, they are having two rings, and cytosine and thymine is having only one ring. How many rings are present? One ring. Okay, so adenine and guanine is having two rings and cytosine and thymine is having how many rings? They are having only one ring. Okay, so that also you have to make a note. Two ring you can understand, isn't it? Here it's clearly given. So how many rings are there? This blue as well as this blue, two rings are present, here only one ring. So adenine and guanine is having two rings, that is the purines are having two rings and pyrimidines are having only one ring. Okay, so this is the structure of guanine, this is the structure of cytosine, adenine and thymine. This is your sugar, the ash one, and the golden one is your phosphate. So like this the nucleotides are arranged. Now you know what is the nitrogenous base. So what is nitrogenous base? Adenine, guanine, thymine, cytosine. We call it as what? Nitrogenous bases, isn't it? Now when nitrogenous bases, adenine pairs with sugar, adenine is pairing with sugar, it form what? It form a nucleoside. When it pairs along with phosphate 3 together, we call it as nucleotide. Okay, so when thymine is pairing with sugar, we call it as nucleoside. When it is pairing with phosphorus, we call it as nucleotides. 3 together, 2 together, we call it as nucleoside. Which two? The nitrogenous base and sugar, nucleoside. When phosphate is also added to it, it becomes 3 compound together, that is nucleotides. Okay, so this is the structure of your DNA. I will be sharing you a worksheet based on the same. Hope so that you have understood clearly, and I want you all to complete the worksheet of this week as well as the last week and send it to me as soon as possible by this Saturday. Let us keep it as a deadline to complete and send. Okay, so just a recap of what we have learned. DNA, double stranded molecule, right handed molecule, double helical structure, twisted ladder. Fine. How many strands are there? 2 strands. They are moving from which direction? 2 strands are there. One is moving from 5' to 3' and another one is moving from 3' to 5'. And when you are saying the diameter, it is 2 nanometer. Between each turn there is 3.6 nanometer. Distance between the base pair of 2 successive rings is what? It is 0.34 nanometer. And in each turn you can find how many nucleotides will be present. Around 10.5 nucleotides will be present. There are 2 groups, one is major and another one is minor groups. And the backbone of the helix, when you are saying, they are made up of 2 things. One is your sugar and another one is your phosphate. That is forming the backbone, and the ladders are made up of nitrogenous bases. It are 4 types: adenine, guanine, thymine and cytosine. Now this purine, when you are saying this purine is adenine and guanine is the purine, which is having how many rings? They are having 2 rings. And thymine and cytosine is pyrimidine and they have only 1 ring in their structure. Okay, and adenine always pairs with thymine, guanine always pairs with cytosine. Adenine and thymine pairing double bond, guanine and cytosine pairing triple bond. So according to complementary base pairing, adenine is pairing with thymine and guanine will always pairs with cytosine, and the number of purine is equivalent to number of pyrimidines, and the bases are attached with one another with the help of a bonding called as what? Hydrogen bonding. With this I wind up for today. Thank you.